Ayun, okay. So, the first law, so bakit ba kailangan nating aralin yung mga batas, 'di ba? Well, hindi naman tayo law student, right? However, syempre, do not forget na kasama sa provision of keeping our license is to avoid illegal activities. So, ibig sabihin noon, if you're going to participate in an organization or a laboratory na illegal, hindi naman siya license, marami siyang mga ginagawa na substandard, di ba? Then definitely, meron kang criminal liability doon. Okay? That's why, kailangan mo ng knowledge. Ano ba yung tama? Ano ba yung mali? So, ito yung mga batas sa Pilipinas that are very important in our practice. So, the first is RA 4688. That is known as the Act regulating the operation and maintenance of clinical laboratories and requiring them to register. Okay, for the same with the Department of Health, providing penalty for the violation thereof and for other purposes. So ito yung batas that requires the clinical laboratory to be established and also to be regulated under the Department of Health. Okay? So ano daw ba yung clinical laboratory na sakop ng batas? So sabi diyan, RA 4688, under diyan ng clinical laboratory. So ano daw ba yon? So these are laboratories in which the body fluids, the tissue secretions, and also from humans and also from animals, 'di ba? Kasama 'yan, kasama yung mga animal laboratory, are determined for are analyzed for the determination for the presence of pathologic organisms, processes, and or condition in person or animals from which they will be obtained. They shall register lahat daw, whether it is a vet laboratory or laboratory for humans, they must be registered and they must be licensed under the Department of Health. And then the one who will supervise that is the Secretary of Health natin. Okay? However, there are some exemption. So exempted dito ang government hospital laboratories doing routine work. Okay? So hindi sila kasama dyan kasi they are part of the services of the government. So under sila directly ng Department of Health. That's why exempted yung mga government hospital laboratories. Now, ang sabi diyan, a laboratory, well, alam natin 'to, 'di ba? Dapat daw merong siyang head, right? At ang head niya is a licensed physician or pathologist certified by the Philippine Board of ay Philippine Society of Pathologists or PSP. Okay? Now, besides that, the Secretary of Health, siya yung responsible to enforce all of this law. Okay? So meron tayong person in charge dyan. The person in charge is the Secretary of Health from the Department of Health. So if anyone violates this act, definitely you will be imprisoned for not less than 1 month, but not more than 1 year, and a fine of not less than 1,000 and not more than 5,000 pesos. So napakababa ng penalties, 'di ba? Kasi hindi pa ito nare-revise. Ito yung mga lumang batas natin na kailangan ma-revise na. Now, okay, so yan, 1966 pa yan. But still, sa hanggang ngayon, pinapatupad pa rin. Kaya makikita mo, napakababa ng fine, di ba? Kasi during 1960s, malaki na yung 5,000 pesos na yan, ha? Yan, those are examples. So that is the RA 4688. Now, ano naman yung mga rules and regulations, 'di ba? That or the implementing rules and regulations that will implement the RA 4688. Doon na papasok si AO. AO or Administrative Order 59, Series of 2001. So these are the rules and regulations governing the establishment, operation, and maintenance of clinical laboratories in the Philippines. So kung paano daw ipapatupad yung 4688, dito siya nakalagay sa AO 59 Series of 2001. Okay, so that is the name of the AO or Administrative Order. So what is the purpose of that one? So class hinighlight ko na dito yung mga importante. So paki-highlight na lang din sa copy nyo. So this handout is part of your module number 8. So you can print that and then please highlight as well yung mga naka-highlight ko kasi ito lang yung importante. Importante rin naman yung iba pero I'm not going to emphasize on that. Ito yung mga importante that you have to know. Okay, so sabi dito, ang purpose of this law is to protect and promote the health of the people to make sure that your clinical laboratory is abiding to the Rules and Standards of the Philippines. Kasi itong ano na 'to, itong AO na 'to, this is all about the Philippine standard. So, ang gagawin diyan meron silang mga basic standard o basic requirement na dapat sundin ng isang clinical laboratory. Okay, so that is the purpose of this administrative order. So, lahat ng sakop nito are all entities performing the activities and functions of a clinical laboratory. So, again, exempted na naman, 'di ba? Hindi kasama ang mga government laboratory. So, they do not include government laboratories doing examinations such as limited acid-fast bacilli microscopy. Ito yung microscopy for Mycobacterium tuberculosis. Yung TB, 'di ba? Yung sakit sa baga, yung TB. Okay, so hindi kasama doon yung mga laboratory specializing for the microscopy lang ng tuberculosis such as the acid-fast bacilli. Malaria screening, di rin kasama, cervical cancer screening, and other services provided as an extension of the government. Okay? And then, ano ba yung function? Well, ito na-discuss ko na to last time, di ba, during prelims, right? Yung classification of function, classification of institutional character, service capability. So, paki-review na lang ito. Hindi ko na siya ulitin na kasi na-discuss na natin yan. And then, under siya sa batas natin, ito yung nasasabi sa batas, these are the different tests and different classification of a clinical laboratory. Okay? Now, an approved permit. So, para ikaw makapagkuha ng permit to construct a laboratory, dapat meron kang design, di ba? Design muna and layout of the laboratory. You're going to submit that to the BHFS. That is the Bureau of Health Facilities and Services. Okay? So, kapag nasubmit mo niya sa Bureau of Health Facilities and Services, they will inspect your layout, your design, kung yan ba ay pasok sa standards. Kapag hindi, you have to revise that. Kapag okay na sa BHFS, then you can now submit an application form. Yung application form, nandoon yung sa baba ng document na to. Meron doong sample ng mga forms. Okay? And then again, no clinical laboratory shall be constructed unless plans have been approved and construction permit issued by the BHFS. Now, ano ba yung mga iba pang requirements, 'di ba, on how to apply or application of permit to construct and license and to operate. So of course, kailangan mo ng application letter, 'di ba? Addressed to the director of BHFS. And then you must have the 4 sets of site development plan and floor plans. Okay? Yan, kasama yan plus the DTI registration. Now, if you're going to renew your license, so let's say okay na, na-inspect na, pasado ka na sa lahat, paano naman ang renewal process ng license? So, application of renewal license shall be filed 90 days before the expiry of the license. So, this is the schedule kung kailan ka pwede mag-apply for renewal of license. So, kapag NCR ka, January to March. Tayo ay, Pampanga ay Region 3, di ba? This one. So, from February to April, ito yung filing ng renewal of licenses. So, bakit natin kailangan mag-file for renewal of license? Kasi class, we have to schedule for inspection. So, whenever we apply for a license or we renew the license to operate, kailangan ma-inspect ng DOH ang laboratory mo. Okay? That's why meron diyang schedule na application and renewal of license. And then dapat, 90 days prior sa expiry makapag-apply ka nga kasi meron pang schedule ng inspection dyan. Kapag nakita ng inspector na meron ka pa mga kulang, at least meron ka pang time para mahabol yung mga kulang. Kasi kung na-expirean ka na tapos marami kang kulang, hindi ka makakaroon ng license to operate. Hindi ka mabibigyan ng lisensya. Okay? Magsasara ang laboratory mo. And then, for the penalties, let's say hindi ka na-comply, meron kang underground na laboratory, the penalty of 1,000 pesos for late renewal shall be charged in addition to the renewal fee for all categories and the application is filed during the next 2 months after the expiry date. So that's why importante na meron kang license to operate. Okay? Kasi kapag expired na siya, underground ka na. And then ito na yung mga punishment mo. Okay? Now, for the inspection naman, sino ang mag-i-inspect? Di ba sabi ko kanina, there will be an inspection, di ba? Prior to approval of the laboratory or during the renewal process. So, during the application na nag-a-apply ka pa lang, may inspection na. Tapos, kapag nag-renew ka ng license, meron pa ring inspection. So, sino gumagawa ng inspection? Si Director of the BHFS or the CHD. CHD stands for Center for Health Development. BHFS stands for Bureau of Health Facilities and Services. So, yung director nila or the representative from that agency shall be the one who will inspect the laboratory. Now, tuwing kailan ng inspection? Ang inspection, it should be with every 2 years. Binabalikan ka or if necessary. Kailan naman yung necessary na yan? Kasi meron tayong tinatawag na inspection na scheduled. Yan, every 2 years scheduled yan, di ba? Meron namang inspection na surprise, okay, or unscheduled, yung biglaan na lang. Pwede yon, pwede na lang gawin yon. Lalo na kung meron mga complaints sa laboratory mo. For example, meron kang clinical lab, tapos meron kang pasyente, nireklamo ka sa Department of Health, di ba? So, si Department of Health, as a due diligence, ang gagawin niya, magko-surprise visit siya. Surprise inspection sa laboratory. Okay? So, yun yung sinasabi na as necessary. Now, for the monitoring, yan, isa pa. Siyempre, hindi naman enough na na-inspect lang, di ba? So that's why meron talaga mga surprise visit. That is part of the monitoring. So the one who conduct that is the director of BHFS or the CHD or the representative. They're allowed to monitor clinical laboratory at any given time. So, surprise visit ito. Hindi na nasasabihin na magpapakita na lang sila doon. Now, issuance of the license. So again, the license shall be issued by the director of the CHD or its representative if your application is meritorious. Ibig sabihin noon, it is compliance with the standards of the Philippines. Now, for the terms and conditions of license, again, nabigyan ka lang ng license to operate if you are in compliance to the standards and all of the other licensing requirements. And do not forget that the license is non-transferable. Ibig sabihin noon, doon lang siya sa laboratory mo. Hindi mo siya pwedeng ibigay sa ibang laboratory. Now, just in case you're going to transfer a location, so nilipat mo, kunyari, meron kang sariling laboratory, pero nag-rent lang kayo doon sa building, 'di ba? And then yung building na 'yon nagkaproblema, so kailangan mong lumipat. So before you transfer to a new location, kailangan ma-inspect muna siya. So sabi dito, the laboratory in its new location shall be subject to re-inspection and shall comply with the licensing requirements. So ulit ka uli ng mga requirements. Magpapasa ka uli. Okay? So any change on the conditions of the license to operate a laboratory shall be reported within 15 days in writing to the BHFS or CHD. So before ka lumipat, 15 days before, dapat mag-notify ka na, nalilipat kami. And then bago kami lumipat, magpapa-inspect ka muna. Kasi hindi po pwede na lumipat ka na tapos nag-operate ka na uli, 'di ba? Kasi nga hindi ka pa na-inspect kung yung bago mong facility is compliant ba sa standards ng Department of Health. Okay? So if ever you have an extension of a laboratory, merong separate license yun. Kunyari, yung business mo nag-grow, di ba? Meron ka ng mga branch. May branch ka sa San Fernando, may branch ka sa Mabalacat, may branch ka sa um, Angeles, 'di ba? 'Yon. So yung mga branch mo, kanya-kanyang license din 'yon. Iba yung branch sa San Fernando, iba yung license sa Angeles. Okay? So ganun siya. Now, the clinical laboratory license must be placed in a conspicuous location area within the laboratory. Ibig sabihin ng class, dapat nakikita siya ng lahat. So kapag pumasok yung mga pasyente mo sa laboratory, yun ang una nilang makikita. So, hindi siya tinatago ha. Dapat talaga nakikita ang license to operate ng clinical laboratory. So, ano ba yung mga violations? Okay? So, the license to operate of a clinical laboratory shall be suspended or revoked by the Secretary of Health upon violation of RA 4688 and this AO. So, kapag meron kang violation sa AO together with the RA 4688, pwede kang tanggalan ng lisensya ng Secretary of Health natin. Okay? So, ano ba yung mga different violations na pwede mong gawin? Well, of course, number 1 dyan yung operation without a certified pathologist. Okay? So, dapat meron kang certified pathologist or registered medical technologist. Okay? Yan yung mga number 1 na pwedeng maging violation mo. Change of ownership na hindi alam ng BHFS. So pag nagbago ka ng ownership, location, head of the laboratory, or any personnel without informing the BHFS or CHD, pwede mo suspend ng license mo. Now, refusal to allow inspection of the clinical lab, possible din. Of course, gross negligence, di ba? Nagkaroon ka ng reklamo from the patient. And any omission, any act or omission detrimental to the public. So, bakit nakalagay dito? Operation of a clinical laboratory without a certified pathologist or without a registered medical technologist. Bakit kaya siya naka-emphasize? Kasi class, noong mga 90s, di ba, konti pa ang medtech sa Pilipinas. Kaya ang ginagawa nila, nagte-train lang sila ng someone para gumawa ng work ng medtech. Tapos, meron silang isang RMT na naka-duty. So, for example, sa lab mo, isa lang yung RMT mo, pero yung apat mong kasama doon, hindi sila RMT. Kung ano-ano lang yung kanilang profession. So, ganun kasi dati. That's why, naka-emphasize siya sa batas ngayon na kailangan meron kang registered medical technologist doon. Okay? Now, so those are the different ways para mag-violate ka ng rules and regulations. So what are the different? So pakibasa na lang to class yung mga investigation of charges or complaints. Ito lang naman yung mga penalties and fines. So hindi naman siya ganun ka-importante. So this one, Section 10, this is all about modification and revocation of license. So a license may be revoked, suspended, or modified in full or in part of any material false statement by the applicant or as shown as the record of inspection or violation. So let's say dinaya mo daw yung mga documents mo. Meron kang pandaraya doon. So, possible na ma-revoke yung license mo, di ba? And then, meron kang violation mismo upon inspection. Hindi ka na-comply sa standards. Kunyari, yung mga machine mo kulang, di ba? And also, yung mga under the rules and regulations of this act na violate mo. So, ganun, yun lang naman yung reason kung bakit masususpend ng lisensya mo. Either, nameke ka, meron ka mga fake documents, or hindi ka na-comply sa standard. Yun lang naman, 'di ba? And then that is the AO. Now this is the technical standards. Ito naman yung kanyang mga implementing rules and regulation. So pagdating sa staffing, so nakalagay sa batas licensed physician, pero talaga ang preferred talaga should be a pathologist. However, konti lang ang pathologist sa Pilipinas. That's why allowed din ang licensed physician as long as nag-training siya for 3 months on clinical laboratory medicine, quality control and laboratory management, and certified by the Philippine Board of Pathology. So, yun ang mga conditions. However, pathologist pa rin talaga yung first choice. Pero konti ang pathologist sa Philippines, di ba? Yun. Now, the clinical laboratory shall employ qualified and adequately trained personnel. So, ano ba yung ibig sabihin ng qualified? It means that your staff are licensed. So, RMT sila. So, kailangan RMT with adequate training. Ibig sabihin noon, nag-undergo ng internship program or any other training pa. Okay? Pwede yon. And then, again, sabi dyan, dapat meron kang sufficient number of registered medtech proportional to the workload. Okay? So, that is one of the requirement as well. Now, for the physical facilities, importante dyan yung space. Dapat meron kang adequate space. Now, the space should be well ventilated, adequately lighted, clean, and safe. And then you should have an enough working space to accommodate the machines, equipment, together with the staff of the laboratory. Kasi kung primary lab ka lang, na napakaliit lang ng square meters nyo, hindi naman po pwede na 10 kayo doon, tapos tambak din ng mga makina, di ba? So kapag ganun, walang adequate working space, pwede kayong hindi bigyan ng license. And then of course, water supply is also very important. Now, these are the measurements ng mga clinical lab. Kapag primary, okay na yung 10 square meters pwede naman. However, for the secondary, 20 square meters ang minimum. But for the tertiary, 60 square meters. Okay? However, separate sa tertiary ang microbiology room. Meron siyang separate na area. Hindi siya dapat kasama sa main lab. So, those are the requirement. Kapag primary lab, 10 square meters lang. Secondary lab, 20 square meters. Tertiary lab, 60 square meters. So, what are the different instruments and equipments na kailangan? So, these are the following. Pakibasa na lang ito, class, hindi ko na siya isa-isahin. Pero, makikita nyo dito, ito yung mga technical standard nakalagay sa batas natin na dapat kung primary ka, meron kang mga gamit na ganito. If you are under the secondary, you must have these materials. If you have tertiary category, you must have all of this equipment. Okay? So ito yung mga makina na required. Now, if you have other laboratory examinations being performed, let's say meron ka mga special services like fertility testing, then dapat meron ka ring appropriate equipment for that. Okay? So it depends kung ano yung mga special tests that you're going to offer. Now, for the glasswares and reagent supplies, nakalagay din siya that you must have appropriate and adequate amount of glassware, reagents, and supplies necessary for the services. It is also important that you comply with the waste management. When you say waste management, this is under the guidance of Department of Health and the local government. Kasi kanya-kanyang LGU, meron din kanya-kanyang guidelines regarding sa disposal of medical waste. And you must be in compliance to that. Now, for the quality control program naman, so meron tayong dalawang klase ng quality control program. You have the internal quality control, you also have the external quality control. When you say internal quality control, these are the actions or process being done inside the laboratory. And all of that should be well documented. So, it is under the internal quality control is the continuous competency assessment program for all laboratory personnel. Ibig sabihin ng class, yung mga laboratory staff, minsan meron silang exam. Pinibigyan sila ng exam ng chief medtech. Ano yung mga exam na yon? Practical exam. So, kunwari, bibigyan siya ng isang slide for CBC. Tapos, si medtech, kailangan niyang basahin yon. Yung kanyang reading dapat tama, dapat pasok dun sa reading ng mga ibang staff din na senior sa kanya. Okay, yun ang tinatawag na competency assessment program. So that is part of the internal quality control. Another thing is that you must also have appropriate and standard laboratory methods, reagents, equipment, and supply. So ibig sabihin noon, yung mga gamit mo dapat within the Philippine standard. Hindi siya dapat substandard. So kung kumukuha kayo ng mga materials sa mga substandard na facilities, definitely, you are not compliant. So when you say standard laboratory methods, ano ba ang basis natin para masabi natin that our procedure that we do in the laboratory is really the standard one? Well, in our practice, we have the CLSI, the Clinical Laboratory Standards Institute, di ba? Sa CLSI class, sila yung agency, sila yung organization pala that develops those different testing standards. Sila yung gumagawa ng mga method. Paano ba mag-test ng glucose? Paano ba mag-test ng cholesterol? Paano mag-identify ng bacteria? Lahat yan merong standard coming from the CLSI. Yun ang ating basis, di ba? So that's why kapag nag-inspect si DOH sa laboratory mo, titignan niya yung SOP mo, di ba? Yung standard operating procedure mo. Titignan niya kung yung procedure mo doon, kapareho ba ng procedure na nasa CLSI. Okay? So, yun ang basis niya para masabi niya na meron kang standard laboratory methods. Now, for the standard reagents and supplies and equipment naman, merong listahan ang Department of Health kung ano ba yung mga test kit, yung mga reagents na approved ng Department of Health para sa Pilipinas. So, hindi po pwede na o-order ka sa China on your own, di ba? Hindi pwede yun. Dapat, yung in-order mo sa China, approve siya ng Department of Health. Approve siya ng FDA din, di ba? May mga ganun reagent na kailangan approve ng FDA. Kasi kung bibili ka ng sa China na underground na kasi mura hindi dumaan sa FDA, hindi dumaan sa Department of Health, makakamura ka talaga. Pero kapag nahuli ka, papasara nila yung laboratory mo. Okay? Another thing is the proper maintenance and monitoring of all of the equipment. So when you say proper maintenance and monitoring, it means that from time to time, the company that supplied the machine, they're going to send an engineer or a representative to conduct preventive maintenance. So yung makina, binubuksan yan, tinitignan kung okay pa yung loob. Pinapractice kung working properly pa yung kanyang mga machineries inside the machine. So that is an example of preventive maintenance. Okay? Ginagawa yan from time to time. So the program shall provide for the use of quality control reference materials. So when you say quality control reference materials, ito yung mga binibili sa industry or the commercially prepared reagents na ginagamit to test for quality control. So, meron tayong tinatawag na positive control and negative control, di ba? Yung positive control, ito yung sample na merong positive result. Kunyari, sample na mataas ang WBC, sample na matataas ang RBC, WBC, di ba? That is the positive control. Tapos, kapag pinasok mo siya sa makina mo, dapat yung makina mo, mababasa niya, matataas yung mga results. Okay? Kapag naman negative control, ito yung specimen na wala, di ba? Specimen na negative ang mga result. So, kapag pinasok yun sa makina mo, yung mga results niya dapat puro negative din. So, that is an example of quality control reference materials. It is commercially prepared. Binili mo siya coming from the manufacturer of your laboratory or of your machines. Okay? Coming from the manufacturer of the machine. Now, you also have what you call the external quality control program. When you say external quality control program, ito yung tinatawag natin na EQUAS, wherein you're going to acquire a specimen coming from another laboratory or the reference lab, di ba? So sabi ko nga sa inyo last time, meron tayong mga different reference lab. Saan ba yung mga reference lab dito? Wala ba dito? Andun pala sa PowerPoint mamaya. So yung EQUAS kasi class, ito yung kumuha ka ng specimen coming from the NRL or the National Reference Laboratory. For example, if you are a laboratory performing test for HIV, 'di ba? Yan. So kailangan mo ng quality control program. Kailangan mong i-test kung yung laboratory mo talaga nakaka-detect ng positive HIV and negative HIV. So ang gagawin mo, kukuha ka ng sample from the SACL, 'di ba? From SACL, yung sa San Lazaro Hospital. They're going to send you the sample and then kailangan i-test mo 'yon. Kapag nag-positive, positive, okay talaga, 'di ba? However, kapag nag-send siya ng specimen na positive pero negative pala, 'di ba? Ibig sabihin may problema sa ano nyo, sa system ng laboratory nyo. So for example din sa CBC or sa ano sa yun, sa CBC, 'di ba? So, kukuha ka ng sample ng CBC sa NRL, sa National Reference Lab. And then, dapat yung mga values ng CBC pasok sa values na nanggaling sa kanila. So, dapat pareho kayo ng resulta. So, that is what you call the external quality control program. So, kung wala ka internal quality control program, wala ka ng external quality control program, hindi ka mabibigyan ng license. Now, for the reporting, so all laboratory reports on various examination shall bear the name of the RMT together with the pathologist. So kailangan daw when you release your results, dapat merong pirma. Sino ang pipirma? The registered medtech who performed the test together with the pathologist na naka-duty during that time. Okay? Now for the recording, ibig sabihin niyan, you're going to keep the records or the medical records of the patient. So there must be a system of recording or keeping the requests and the results of the specimens. So, sabi dyan, all records of the laboratory shall be kept in file for at least 1 year. Kapag more than 1 year na, pwede ka na magtapon. Except for the records of anatomic and forensic pathology. Kasi ito yung mga may kinalaman sa krimen, di ba? Sa mga criminal case, yung mga forensic na yan, di ba? They shall be kept permanently in the laboratory. Yon. So, yung permanent file is the anatomic and forensic pathology file. However, yung mga normal lang, 1 year lang yan. Tinatapon na. Now, for the laboratory fees naman, hindi pwede ang overcharging. So, nakalagay dito, kung maniningil ka, yung rate mo, it should be within the range of the usual fees prevailing at the time. So, wala silang binigay na standard fee. Kasi nga, from time to time, nagkakaroon ng inflation, di ba? So, nagbabago talaga yung mga singil sa mga laboratory. However, dapat pa rin hindi ka overpricing. Dapat enough lang siya to cover the cost of testing. So, kung magkano yung puhunan mo sa mga reagents na ginamit mo, and quality control of various laboratory procedures. And of course, the professional services rendered to the patient in the performance of special procedure or examination shall charge separately and not included in the laboratory fee. So if you have other services, ano ba yung mga other services na pwedeng ibigay sa pasyente? Example of that is the home service, 'di ba? Yung home service, ibig sabihin ng home service, merong medtech pupunta sa bahay mo para kumuha ng dugo. So, ang babayaran ng pasyente, yung laboratory fee. Yung laboratory fee, ito yung mga examination sa kanyang dugo tulad ng CBC, clinical chemistry test, yan ang laboratory fee. However, yung transportation fee ng medtech together with the food of the medtech, separate ang bayad doon. Kasi kung malayo yung medtech, 'di ba? 'Yun. So, dapat separate 'yun. Depende 'yun kung magkano yung kung gaano kalayo yung pinanggalingan and kung magkano yung naging transport cost. Now, this one are the files. Ito lang yung mga files na related sa application. So, meron siya ang mga forms. Yan. So, tignan nyo na lang siya. Hindi naman kailangang i-memorize. So, this one, ito yung mga test na pwede mong i-offer sa laboratory mo. Yan. For the special urinalysis, these are the following tests. So, pakibasa na lang. Hindi naman kailangan i-memorize pa ngayon. Kasi itong mga test na yan, you're going to learn that pagdating ng MLSP2. Doon nyo siya aralin. Okay? Yan. So, it is just for this time, mga nice to know pa lang siya. Yon. So, that is the technical standards. So, sabi dito, for private laboratories, Certificate of Registration of Business Name from the Department of Trade and Industry, kailangan din siya as a requirement. Certificate from the Philippine Board of Pathology, PRC Board Certificate of the Medical Technologist, notarized statement that the applicant has complied with all the business requirements under the existing laws, location map, and floor diagram. Okay, so these are the basic requirements. Okay, so that is the AONRA 4688. Now, for the next topic, I'm going to talk about the clinical laboratory, the different sections in the clinical lab. Okay? So I have here the different sections in the clinical lab. Ayan. Okay. So the first section is the clinical chemistry. So I have here a list of tests being offered under clinical chemistry. So ano ba ang focus ng clinical chemistry section natin? Testing of blood and other body fluids. What are you going to test in the blood? All of the essential soluble chemicals found in blood and urine. So those, ano bang example? Example are the blood sugar, the hemoglobin A1C, 'di ba? Total cholesterol, triglycerides, HDL, LDL. So ito yung mga test mo for your lipid profile. For the BUA, BUN, creatinine, electrolytes, potassium, calcium, magnesium, those are example of electrolytes. You have the total proteins, you have the bilirubin, troponins, CRP. You have the CK, amylase, lipase, GGT, AST. You also have the hormones, the TSH, T3, T4. And you have the iron, ferritin, TIBC. These are the tests for anemia. And you have their drug testing, therapeutic drug monitoring, and quality control in waste management. So as of now, class, sa MLSP1, ang kailangan ko lang gawin is banggitin sa inyo itong mga test. Okay? I'm not going to explain kung ano yung mga clinical significance niya. Kasi that will be discussed to you next semester sa MLSP2. Itong isang PowerPoint na yan, class, ilang meeting ba ito sa MLSP2? Parang 2 meeting ito. Mahaba yan na discussion sa MLSP2. Okay? Yon. So, yung mga significance niya sa MLSP2 na. Sa ngayon na MLSP1, babanggitin ko lang yung mga test. So, dapat kapag meron ako nasabing test, let's say sodium, dapat alam nyo na under siya ng clinical chemistry. Okay? FBS, under ng clinical chemistry. Amylase, under ng clinical chemistry. So, yun pa lang as of now. Okay? Hindi nyo muna aalamin kung ano ang clinical significance ng mga yan. Pagdating nyo sa third year, isang buong semester, actually, hindi pala, 2. 2 semester nyo aaralin ito lahat. So, napakahaba niyan, Yun. Okay? So, since first year pa lang kayo, paunti-unti muna, introduction muna ito, Yan. So, please be familiar with the different tests conducted in the clinical chemistry section. And then, I have here an example of a fully automated system in the clinical lab. Sana mag-play siya. Nag-play ba to? Ayaw niya mag-play. So, ipakita ko na lang sa inyo sa video. Kasi I want you to see kung ano ba yung ginagawa sa clinical chemistry. Habang sample yun. Wait lang. Alinity. Wait. Abot. Ayun, alinity. Yan. So this one, ito yung example ng makina or example ng trabaho na ginagawa sa clinical chemistry. Sa face-to-face kasi, dapat pupunta kayo sa ospital. Since lockdown tayo, di ba, hindi pwedeng pumunta sa mga laboratory. I'm just going to show you a video kung ano yung nangyayari sa ospital.
Delivering unprecedented integration across your laboratory. Alinity has the power to elevate your performance with increased operational productivity and greater flexibility. This is the Alinity I, and this is the Alinity C, each delivering maximized throughput and load-up capacity with identical footprints. All this power is packed in a more compact size, thanks to an innovative stacked reagent and sample carousel design. As testing volumes increase, these systems can scale to meet your needs, combining up to 4 modules in multiple configurations, and can connect to our open automation system. Alinity is built around you, offering efficiency and streamlined processes.
Okay, so itong machine na to, makikita nyo, ano na siya, multi-system na siya. Ibig sabihin nun, meron kang part na pwedeng gumawa ng test ng clinical chemistry. Pwede mo siyang i-integrate sa mga tests for immunology and serology. Pwede mo siyang integrate sa mga tests for hematology and coagulation and other tests for urinalysis. Pwede yun. So, integrated yan connected sila. Ito yung tinatawag natin na fully automated system. Na ang ginagawa mo, pinapasok mo lang yung specimen and then siya na yung bahala. Kunyari, from clinical chemistry, meron pa siyang test sa immunology. Siya na bahala magpasa doon. 'Di ba? Integrated siya, connected siya. Meron yung mga robot arms that you're going to see. Napakita niya.
Offering efficiency and offer an intuitive and easy to use experience into any
Like this. Ayan. Ito yung specimen. Pinapasok mo lang. Tapos merong robot arm. Yan.
And automatically routes samples and reagents to the correct location. To further increase your walkaway time, calibrators and controls can also be stored on board the system. Chemistry testing on our Alinity C with world-class Six Sigma performance is carried out in 187 glass cuvettes, utilizing smartwash technology to minimize sample-to-sample carryover. For even greater efficiency, electrolytes are performed via our integrated chip technology, a single module designed to deliver an impressive 60,000 determinations with CVs of less than 1%. On our Alinity I, immunoassay testing is further enhanced through our patented ChemiFlex chemiluminescence technology. Prioritizing samples is now even easier. You can either press the Stat button, pre-program multiple lanes, or even prioritize through the automation system. Helping avoid potential repeat testing and downtime, error-proof design elements prevent loading solutions and reagents in the wrong position. Thoughtful placement of the sample probe accommodates processing from within the module or from the automated track. The innovation doesn't stop here. Our automation solution is designed to give you a whole new level of confidence. Maximize uptime with intelligent car technology. Easily support any layout with interlocking track components. Optimize your lab floor space with multi-floor transport and elevated track segments. The Alinity CI series, powerful innovation from Abbott that simplifies diagnostics.
Ayan o, yung mga test tube, sila yung gumagalaw. Sila na yung bahala pumunta. Napaka-high-tech. Ayun, that's an example, di ba? So, kapag ikaw RMT na, nagtatrabaho ka na in the hospital, part din ng trabaho mo ay alamin kung paano gumagana yung mga makina na yon. That is also part of your job. So, kapag nag-work ka na, gagawin dyan, you'll be given a training kung paano gamitin yung makina, bubuksan nyo yung makina. Titignan mo yung mga parts dyan, tsaka yung mga basic things that you have to know para malaman kung working properly ba yung machine. Okay, ganun siya, ha? Ganun kapag ikaw ay isang RMT. And then, since nakita nyo, technology yun, di ba? Since yan ay technology, of course, hindi pa rin talaga nawawala sa atin ang mag-conduct tayo ng mga trainings and seminars or mag-attend sa mga trainings and seminars. Kasi nga naman, laging merong bagong innovation. Tulad niyan, yung Alinity. Ngayon lang yan. Noong nagtatrabaho ako, wala pa niyan. Ang meron sila yung architect, di ba? Which is also something like that, pero walang mga robot na gumagalaw. Iba. So, iba na ngayon. Okay? Now, under naman ng microbiology, all about study of bacteria and fungi ito. Pwede rin ang mga viruses. However, separate ang laboratory ng virology lab. Kasi nga naman kapag viruses, highly infectious, di ba? Sobrang liit niya kasi. So, madali siyang kumalat. Okay? Kaya nga naman kapag meron kang virology lab, hiwalay yan. So, ano makikita mo on the microbiology? Pwede kang mag-conduct ng microscopic examination like this one. Ito yung mga bacteria. Nakikita mo, ito yung dugo mo. This is your blood kasi meron kang mga white blood cells. Ito, white blood cells yan. Tapos makikita mo, meron mga bacteria dyan sa dugo mo. Ibig sabihin yan, meron ka ng sepsis, di ba? Meron kang infection sa blood, which is actually delikado. Now, for this one, this is a test on performing what you call bacterial culture. Ibig sabihin noon, pinapatubo mo yung bacteria sa mga petri dish. Kapag tumubo na siya, ito yung kanyang itsura. Ito, mga bacteria yan. This is what you call the bacterial culture. And then this is also the fungal culture. Kapag fungal, yung mga amag, yung mga fungi, like this one. Yan, pinatubo mo siya sa petri dish. Ito na ang kanyang itsura. Those are the fungi. Okay? So that is what you do in the microbiolab. Lab. Ano pang ginagawa sa microbiolab? Again, culture and identification of the bacteria or fungi, gram staining or other stain. Tulad ng ginawa dito, di ba? Meron kang blood tapos in-stain yung mga bacteria. And of course, antibiotic susceptibility testing. When you say antibiotic susceptibility, you're going to test what are the effective antibiotic that can actually kill the bacteria. So itong bacteria na pinatubo mo, iti-test mo yan against different antibiotics. For example, iti-test mo siya against levofloxacin, ciprofloxacin, ampicillin, di ba? Tapos titignan mo kung alin sa antibiotic na yun yung nakakapatay sa bacteria. Kasi yun ang ibibigay ng doktor sa pasyente. Kasama rin dito ang quality control and waste management program, of course, because you are dealing with infectious waste. And also, infection control and biosafety. When you say infection control and biosafety, this is a program inside the microbiolab that aims to contain all of the infectious microorganism inside the laboratory. Kasama din ang proper waste management niya. Kasi kapag yung mga mikrobyo na yan nakalabas sa microbiolab, kunyari napunta sa hima, napunta sa clinical chemistry, pwede magkaroon ng sakit yung mga tao sa hima and clinical chemistry. Bakit? Kasi hindi sila prepared for that microorganism. Kapag kasi nasa microbiolab ka, meron ka mga protection doon, meron kang gear, di ba? Meron kang PPE, minsan naka-hazmat suit ka pa, depende kung ano yung particle na yung microorganism na inaaral nyo during that time, di ba? Yun. So, however, yung mga staff na nasa clinical chemistry, wala silang equipment na naka-ready. Kaya nga naman kapag lumabas ka ng microbiolab, tapos pumunta ka sa clinical chemistry, nakipagkwentuhan ka, so, however, hindi ka pa naghuhugas ng kamay, hindi ka nagpapalit ng lab gown, dala-dala mo yung mikrobyo from microbio, dinala mo ng clinical chemistry. That's an example of infection control and biosafety. So, dapat meron kang program on how to contain that. That's also under microbio. Now, for the viruses, ano yung mga test na ginagawa? They can perform cell culture. Kapag cell culture, pinapatubo mo rin yung viruses. However, hindi siya sa petri dish siya, hindi siya tulad nito. Ito kasi, culture iyan para sa bacteria. Ito, culture for fungi, kaya sa Petri dish. Okay? However, kapag cell culture yan, you're going to culture the viruses inside living cells. So, meron ka mga nakaredy na cell line, doon mo pinapatubo ang mga viruses. Now, besides that, you're also going to perform microscopy. However, it is different from this one. Ito kasi bacteria, malalaki yan. Kaya madali lang siya makita under the microscope. Kapag viruses, it is very small, very minute, that you need a very powerful microscope such as the electron microscope para makita mo siya. Kasama din doon ang immunodiagnosis. Ibig sabihin niyan, we're going to detect the viruses via antibody or antigen testing and also PCR testing. But for the PCR, this is done under the molecular biology section. Now, this is an example of automated bacterial culture. So, pakita ko na lang din sa inyo to. Wait lang. Dapat kasi, uutusan ko kayong pumunta sa isang ospital. Ganun kasi ang week na ito. Kapag kung face to face ganun ang gagawin. Kaso nga lang, lockdown tayo. Kaya, tignan nyo na lang dito kung papaano ang ano bang title na to? Microbiology. Okay, so ito na lang yung example natin. So this is an example of a microbiolab.
Has been used for decades, not only to save energy and materials, but also to improve
Yan.
quality and accuracy in healthcare, especially here in the laboratory. We've used automation for blood tests for years now. It was really great results. But in microbiology, we're a little bit different. We have to identify what microorganisms or bugs. We have to make sure that the right patient gets the right treatment for the right condition. The process of running these tests and reading these results has been a very manual process.
So ito yung mga manual process na ginagawa. So nakikita nyo, meron siyang mga petri dish dyan, tapos meron siyang ginagawa na streaking method. Okay, that is the manual. So meron ng automated niyan. Makina na ang gumagawa. This one.
Today's microbiology labs face many challenges, including increasing volumes, cost containment pressure.
Okay, so i-forward natin. Yan, ito yung mga bacteria. Ayan na, yung petri dish. Yan. Ito, binabasa siya ng machine, automatic. Yan.
We're the second in Texas and the very first in Harris.
Ito yung mga petri dish, pinapasok siya automatically. That one, yan.
All of this automation sounds great, and it really is an amazing system. But the real impact is what it will mean to us.
Microbiology automation.
So may kita mo yung petri dish, 'di ba? Siya na mag-isa gumagalaw, 'di ba? Yan, yung mga microorganism. Yan.
It's really exciting to be on the cutting edge
Okay.
of this kind.
So yan ang example ng automated system sa microbiology. By example, pwede sa inyong bigay. Anyway, actually, mas maganda talaga pupunta kayo sa ospital para makita ito. However, bawal talaga. So that's just an example, 'di ba, ng mga automated system natin sa laboratory na ginagawa. Now, so continuing with the lesson. So after the clinical microbiology, you also have here hematology and coagulation. So for the hematology and coagulation, ito yung section that studies the blood cell. So makikita mo, these are the RBCs. Itong mga pink na yan, those are the eosinophils. Itong mga merong bilog-bilog na parang segmented, those are the neutrophils. And then this one, yung may grayish, that is the monocyte. And then this one, yung bilog lang, this is the lymphocyte. So ito yung mga blood cells mo. Okay? So, hematology is all about enumeration of blood cells in the body and other body fluid. So, part of the test is the CBC or the complete blood count. Also, your hemoglobin and hematocrit. The WBC differential count. Ibig sabihin niyan, itong mga cells na yan, binibilang mo siya. Okay? And then the blood indices or the MCV, MCH, MCHC, RCW. And also the coagulation studies such as PTN, APTT. So again, lahat ng significance will be discussed to you during your third year, and also the MLSP2. So next is the clinical microscopy. Now, for the clinical microscopy, kasama dito ang urinalysis and fecalysis or stool analysis. So dito sa picture, these are examples of crystals found in the urine. Kasi yung mga crystals na yan, meron silang kanya-kanyang clinical significance, lalo na yung mga abnormal crystal. Na kapag nakita mo daw itong mga lucine crystals na yan, meron kang problema sa liver. Okay, that's an example. Now, this one, this is the region strip test na ginagawa sa urinalysis. Ang ginagawa mo, itong mga strip na yan, sinasawsaw mo siya sa urine, and then yung kanyang mga color reaction, kino-compare mo siya sa standard para basahin yung mga different chemical tests for the urinalysis. And then this one, these are example of parasites, ova, yung mga itlog ng parasite. So just by taking a look at the ova, you can actually identify what is the parasite found in the patient. Kasi kada isang parasite, meron siyang distinct na itlog na sa kanya lang. Ganun kagaling ang nature. So, hindi pare-pareho mga parasite. Kanya-kanya silang itsura ng itlog. Okay? Now, for the urinalysis, meron kang tatlong part dyan. Meron kang physical examination where you're going to check the color, clarity, and specific gravity. And you also have the chemical examination where you're going to check the glucose, protein, leukocyte esterase, bilirubin, blood ketones, urobilinogen, pH, and nitrite. So, ito yung chemical examination niya. Tinitignan mo yung mga different chemical tests na makikita mo dito. And of course, for the microscopic examination. So example dyan, ito, nakikita mo yung mga crystals under the microscope. So besides the crystals, you're also going to examine for RBC, WBC, squamous epithelial cell, urine cast, urine crystals, bacteria, parasites, and mucus. Now, for the fecalysis, so sabi ko nga kanina, this is all about identification of the parasites. So you can identify the parasites by taking a look at the ova, like this one. So ito, this is the ova of the ascaris. This is the ova of the trichuris tricura. And I think these are necators or yung mga hookworms nyo. Yung iba, hindi na ako masyadong familiar. Okay? Kasi nga naman, di naman ako parasitologist. But yung mga basic natin, nakikita ito, yung mga ascaris, trichuris, and the hookworms. Okay? Yun. So, next is the blood or the blood bank or immunohematology section. Now, for this section, again, ito yung section that will deal with all of the process before transfusing the blood unit. So, this is all about blood transfusion. Ano yung mga process na ginagawa bago isalin ng dugo? So, this is an example of the blood bag that you're going to process in the lab. For the cross-matching, actually, gel method na tayo. Dati kasi tube ang ginagamit, pero ngayon gel na like this one. Or if you want, pwede naman ng automated system like this one. Okay, meron ng makina that will perform the blood typing and cross-matching for you. And this is an example of blood typing. So kapag daw meron kang reaction sa anti-A, and then meron kang reaction sa anti-D, you are A positive, di ba? This one, anong blood type nito? B positive. Kasi meron siyang anti-B reaction and meron siyang anti-D reaction. So, yung kanyang RH is positive. So, B positive yan. Ito naman, meron siyang reaction sa A, meron din reaction sa B, meron reaction sa D, kaya siya ay blood type AB. This is blood type A, blood type B, blood type AB. Yan. Ito naman, walang reaction, blood type O. O yan. Okay, walang reaction siya. Now, for the test under the blood bank, again, you perform blood typing, compatibility testing or cross-matching na tinetest mo kung yung blood bag ba ay compatible sa pasyente, and also antibody screening. Under din dito ang blood donation, screening of the blood units against different infections, and also storage of blood. And then, you're also going to process the blood component. Tulad nito, this is a pack RBC. Pack RBC yan class. Okay? Kasi nga, may kita mo RBC na lang yun nandyan. So, kung hindi man pack RBC, meron ka ring whole blood. Kapag whole blood, kasama pa yung plasma. Meron pang fluid. For the platelet concentrate, only the platelets. For the fresh frozen plasma, only the plasma. Wala siyang kasamang blood. Okay? So, that's an example of the different blood components. And all of that is under the blood bank or immunohematology section. Now, next is the immunology and serology. So, anong example ng mga test na ginagawa sa immunology? Yan, yung antigen test. Antigen and antibody test for COVID-19. That's an example. Ano pa, yung mga test like this one, this is the ELISA test. This is a test performed to check if the patient have hepatitis B. This is also good for hepatitis C diagnosis. Pwede rin. So you can diagnose hepatitis B, syphilis, dengue, hepatitis C by performing ELISA and RIA test. Okay? So this is also an example. Meron ka mga test kit to detect antigen and antibodies for COVID-19. But this test, mukhang ano lang to. Mukhang this is an antigen test. Antigen test lang ito, class. Yan. Kasi ano lang siya, control tsaka test lang. So, antigen test yan. Kapag dalawang line, ibig sabihin positive. So, next is the histopathology or cytology. So, nabanggit ko sa inyo last time na yung mga tissue niluluto yan. Saan sila niluluto? Dito, sa makina. You have here what you call the autotechnicon. So, yung mga tissue, pagkatapos mo siyang hiwa-hiwain ng maliliit na pieces, ilalagay mo siya dito. Yan, sa machine na yan. And then yung machine siya na yung bahala mag-process or magluto sa tissue. So, ang ginagawa niya sa tissue, nilalagyan niya yan ng alcohol, nilalagyan niya yan ng xylene, nilalagyan niya lang ng mga benzene, kung ano-ano mga chemicals pa para mag-harden and soften and dehydrate yung tissue. Kapag dehydrated na and soften and hardened na, then lalabas na yung tissue, lalagay mo na siya sa paraffin. Actually, nakalabas siya na sa paraffin na, 'di ba? And then after that, kapag nailabas na yung tissue, then you're going to prepare the slides. So, yung tissue na yan, kakat mo pa uli, ilalagay mo siya sa slide, then you're going to stain the tissue. So, ano yung itsura ng stained tissue? Like this one. Ito na yung itsura ng mga stained tissue. So, yung mga picture na pinapakita sa libro nyo, ng mga pictures ng mga tissue like this one, tayo ang gumagawa niyan. Tayo ang nagkakat nito. Okay? Tayo nag-i-stain yan, class. Pwede rin yung frozen tissue section. And also, all of that is for cancer diagnosis. So those are example of histopathology or cytology test. Now, for the immunohistochemistry naman, this is a part of histopathology combined with clinical chemistry. So anong ginagawa dito? So this is an example of a stain coming from the histopath. Ito yung normal na histopath stain. Diba? Like this one, ito yung kanyang mga stain na ginawa. However, sa immunohistochemistry, hindi lang normal stain ang ginagamit. Gumagamit siya ng mga fluorescence or fluorochrome. Like this one. Yung mga fluorescent na yan, fluorescent dye na yan, meron siyang kasamang antibodies. Yung antibodies niyon, didikit siya doon lang sa specific target cells niya. That's why, magkakaroon ka ng mga fluorescence dito. For example, you're going to take a look kung yung pasyente meron siyang cervical cancer, di ba? Nakita mo, yung kanyang tissue, mukhang may cervical cancer, pero hindi ako sigurado. Hindi tayo sure kung may cancer talaga yung pasyente, pero mukhang may problema. So, pwede mong gawin yung tissue na yan, ilagyan mo siya ng antibody with fluorescent dye. Ibig sabihin noon, yung antibody niyan, specific siya na didikit lang siya doon sa mga cells na merong cervical cancer. Kapag dumikit siya doon, mag-form sila ng reaction with the enzyme, releasing fluorescence. So tulad nito, may mga fluorescence. Ibig sabihin niyan, positive yan. Kasi may nakita kang fluorescence, di ba? Yun. So ito, like this one, positive yan kasi merong fluorescence. Ito, may fluorescence, kaya positive din. Okay? So, yun yung purpose of performing histochemistry. Kasi nga naman, yung mga normal na histopath test, tulad yan, yung mga tissue examination, mahirap siyang tignan, di ba? Paano mo makikita dyan yung cancer? Alin dyan yung cancer? Di ba? Lalo na kung hindi ka properly trained, right? Kaya nga naman, pwede kang gumawa ng immunohistochemistry to confirm. Kasi kapag nag-bind yung antibody dyan sa cancer cells, tapos nag-release siya ng fluorescence, definitely, positive na nga talaga. So that is the immunohistochemistry test. Yan, this is also another example na ito yung normal na tissue niya, normal staining, tapos kapag may fluorescence itong kanyang itsura. Yan, nag-fluorescence na, di ba? This one, yan. Ito yung mga affected area niya, yung merong fluorescence. Okay? Ito yung kanyang mga normal staining lang. Ito yung merong fluorescence. Yan, that's an example of immunohistochemistry. Now, for the PCR technique or the molecular biology and biotechnology, again, they test for the DNA, RNA, and perform the polymerase chain reaction. So, this is the machine that is being used in the molecular biology lab. Meron kang PCR thermal cyclers like this one. Sana face-to-face tayo, no, para nakapag-tour kayo sa mga ospital. Anyway, darating din naman yan sa tamang panahon. Okay? So, ayun. So, laboratory testing cycle, meron tayong tatlong phase of the laboratory test. You have the pre-analytical, analytical, and the post-analytical phase. When you say pre-analytical, ito yung mga variables that you perform before the actual testing of the specimen that can actually affect the results of the test. For example, CBC, 'di ba? Nag-test ka ng CBC. However, during blood collection, ang ginawa mo is yung blood nung nag-mix ka ng blood sa test tube, sobrang bilis like this one, ganun ang pagka-mix mo. So ang nangyari, yung mga red cells ng pasyente nasira na kasi nga yung pag-mix mo mali. Ano ba ang proper mixing? Paano ba ang proper way on to mix the blood? Dapat like this one, figure of 8. And then dahan-dahan lang siya. Okay? So isasalin mo yung dugo sa test tube tapos figure of 8 lang ang pag-mix. Pero since hindi ka marunong, ang ginawa mo, kunyari nagmamadali ka, tsaka ewan ko, ano lang, hindi mo lang gustong gawin yung figure of 8 kasi matagal. Ang ginawa mong pag-mix ganito, yan. Mabilisan kasi nagmamadali ka. So, yung ginawa mong action na yon, mali yun. So, anong nangyari, yung mga red cells ni pasyente, nagputukan. Kaya, noong nag-test ka ng CBC, ang baba ng resulta ng red cells. Okay? So, that's an example of pre-analytical. So, kasama sa pre-analytical ang receipt of the laboratory request. So, kunwari, nakalagay sa request ng pasyente, clinical chemistry and CBC. However, hindi mo nakita yung CBC. Ang nabasa mo lang, clinical chemistry. Kaya nung nag-collect ka ng blood sa pasyente, ang nakuha mo, for clinical chemistry lang, walang for CBC. So, magkakaroon ka ng failure of test. Hindi mo magagawa yung CBC kasi wala kang specimen. Okay? Another one is patient preparation. So, for example, ang test niya is fasting blood sugar. 'Di ba? So, dapat naka-fasting si pasyente for 8 hours. However, si pasyente, hindi niya talaga matiis. Noong 12 midnight, nagising siya, kumain siya. Tapos, nung tinanong na siya ng medtech, "Ma'am, nag-fasting ka ba? Hindi ka ba kumain ng gabi?" Hindi po, nagsinungaling si pasyente, 'di ba? So, since nagsinungaling si pasyente, pag lumabas na yung resulta ng FBS niya, sobrang taas. Kasi nga, kumain siya ng 12 midnight. 'Di ba? Yun. Eh, dapat di na siya kakain nun eh. Okay? So, yun ang example of patient preparation na part ng pre-analytic. Another thing is the specimen collection, 'di ba? Yung diniscuss ko kanina. Nung nag-collect ka, galit na galit ka sa pag-shake, shinake mo ng gano'n. So, nasira yung mga red cells ng pasyente, 'di ba? Specimen transport, yan. For example, kulang kayo ng gamit sa laboratory na ubusan. So, kailangan mo dalhin yung specimen sa kabilang laboratory. So, ang ginawa mo, nagmotor ka, dala-dala mo lang yung test tube, 'di ba? Nakamotor ka. Traffic, 'di ba? Ang tagal-tagal mo, traffic kasi sa EDSA or sa kung saan man, hindi ka nakarating agad sa laboratory, na-delay ka ng 4 hours. So, pagdating doon sa pangalawang laboratory, definitely, panis na yung specimen, di ba? So, dapat kasi ang ginawa mo, yung specimen, nilagay mo siya sa cooler na merong ice para ma-preserve yung specimen. Just in case ma-traffic ka on the way, hindi siya mapapanis. That's an example of specimen transport error. Okay? So, lahat ng mga variables na yun, lahat ng error na yun, they can actually interfere with the results. Now, for the analytical part naman, ito yung actual testing na ng blood. So, ano yung mga variables dito that can affect the result? Unang-una yung equipment, di ba? If the machine is not working properly, so sira-sira yung mga machine nyo, di ba? Yan. Instrument, yung mga ginagamit nyo sa lab, hindi na siya up to par to the standard, di ba? Yung mga reagents use nyo, substandard reagent, binili mo lang sa underground sa China, mga hindi authorized ng FDA, di ba? Yan. Your quality control program, hindi siya pasok. Definitely, yung mga result mo maapektuhan. Okay? Kaya dapat lahat yan ay within the standard. And also the post-analytic. When you say post-analytic, this is a process on how you're going to transmit the test result. So, saan mapupunta yung result? Dapat mapunta siya sa pasyente and also sa doctor. Kung necessary, pwede rin naman sa nurse station kung admitted. So, yung mga laboratory ngayon, automated na sila. Meron silang system na ini-email nila yung result sa pasyente. Pwede naman yon. Or kung makaluma pa rin, pupunta mismo sa pasyente sa laboratory para kunin yung kanyang resulta. And then also, make sure that the turnaround time is always followed. So kapag stat yan, dapat 'di ba less than 1 hour, right? Or 30 minutes lang meron ka ng result. Kasi nga stat siya, emergency. Pero kapag yan ay routine lang, hindi naman siya emergency, kinabukasan dapat magbigay ka na ng resulta. So kapag sinabi mo sa pasyente na ibibigay ko po ang resulta 8 ng umaga, dapat 8 ng umaga nandun na talaga yung resulta. Okay? That is part of the post-analytic. So ano pa yung mga pwedeng maging error sa post-analytic? Number 1, yung medtech, 'di ba? So sabi ko nga kanina, yung mga test ginagawa siya sa machines, 'di ba? However, kailangan pa rin ng medtech to make sure na yung mga result ay tama. So upon printing of the result coming from the machine, the medtech should double check if the results doesn't have any flags, 'di ba? Kapag walang flag, okay lahat ng result, then okay na, i-type mo na yung result, i-release mo na sa pasyente. And then you must also sign the result and then the pathologist will sign the result. Pero kung nakita mo may error sa resulta, may mga flags, maraming problema sa result, then hindi mo pwedeng i-release agad yung result. Kailangan mo mag-double check. You can repeat the test or you can verify with the diagnosis. Okay? Kasi kung talaga meron, kunyari, meron talaga siyang problema sa kidneys, definitely, matataas talaga yung result niya when it comes to the kidney function test. Okay? That's an example on how to double check the results. Now, for the quality assurance, so nasabi ko na to kanina, right? Ito yung mga ginagawa nyo to make sure that the results are correct, right? So, meron kang internal quality assurance na day-to-day activities mo. Meron ka rin external quality assurance. So, in the Philippines, you have here the different NRL. Ito yung mga National Reference Laboratory. For hematology and coagulation, the reference lab is NKTI. For microbio and parasitology, the reference lab is RITM. For clinical chemistry, the reference lab is the Lung Center of the Philippines. For drug of abuse and toxicology, that will be the East Avenue Medical Center. And for the HIV, Hepa B, and Hepatitis C, that will be for the SACEL or sa San Lazaro. So ito yung mga different NRL in the Philippines or the National Reference Laboratories. Oh, yun na pala yung lesson. So that is the lesson for this week. That is all about the clinical laboratory law. So do you have any question about the discussion?